Bismillahirrahmanirrahim. Today's my lecture is about photochemicals. The light sensitive photochemicals is found in the outer segment of rod and cone. In case of rod, this photochemical is called rhodopsin or visual purple, which is present in membranous discs located in the outer segment of rod cell. In case of cone, it is one of three color photochemical, usually called simply color pigment, which functions almost exactly the same as rhodopsin, except the difference is spectral sensitivity. Both rhodopsin and color pigments are conjugated protein. They are incorporated into the membranes of discs in the form of transmembrane proteins. The concentration of this photosensitive pigment in the discs are so great that the pigment themselves constitute about the 40% of entire mass of outer segment. This is the figure of rhodopsin. Rhodopsin is made up of a protein called opsin and a chromophore. Opsin present in rhodopsin is known as scotopin. Scotopin is found in the membrane of rod discs and made up of 90% of total protein in this membrane. Opsin is a part of large family of G protein coupled receptor. On the other hand, chromophore is a chemical substance that develops color in the cell, which is present in rod cell is called retinal or retinol or retinin or retinin 1. It is also called 11-cis retinal. Retinin 1 is present in human eye. This is different from retinin 2, which is present in the eyes of some animals. Significance of 11-cis form of retinal is that only in this form it combines with scotopin and synthesizes rhodopsin. Retinal is derived from food sources not synthesized in body. It is derived from carotenoid substance like beta carotene, which is present in carrot. As an example, because of the importance of vitamin A in the synthesis of retinal, its deficiency produces visual abnormalities. One rod cone contains 10 billion rhodopsin molecules, which is tightly packed in the disc membrane, which ensure an optimum photon capture rate. When light is absorbed by rhodopsin, decomposition of rhodopsin starts. It is split into retinin and protein called opsin. Through various chemical reactions, this occurs. Rhodopsin absorbs all visible wavelengths so that rod provides vision only in shades of gray by detecting different intensities rather than color. When retina is isolated and examined in dark, rod appears red because of rhodopsin. During exposure to the light, rhodopsin is bleached and color becomes yellow. Photopigments of cone. It is three types, namely porphyrosin, iodopsin, cyanopsin. Only one of these pigments is present in each cone. Photopigment in cone cell also is a conjugated protein made up of protein and chromophore. Protein in cone pigment is called photopsin, which is different from scotopsin, and chromophore of cone pigment is retinal or 11-cis retinal, which is present same as rhodopsin. All the color we perceive results from a mixture of nerve signal from these three types of cone. Each type of cone pigment is sensitive to a particular light and maximum response is shown at a particular light and wavelength. This is the table taken from Sambolingam book. In case of rod, the peak sensitivity is about 505 nanometer. The details of responses of cone to light are probably similar to those in rod. Light activates retinal and this activates a cone transducin, which is a G protein that is different somewhat from rod transducing. Cone transducing in turn activate phosphodiesterase, which catalyze the conversion of cyclic GMP to 5-GMP, which causes closure of sodium ion channel between the extracellular fluid and cone cytoplasm. So, decrease in intracellular sodium ion concentration and hyperpolarization of cone synaptic terminal. This is all about photopigments of rod and cones. Thank you for your patience hearing.